Assalamualaikum warahmatullahi wabarakatuh, and a very good day to my classmates and also Dr. Akmal as our biology lecturer. So today my group is going to present the topic 20.3, that is bacteria. Bacteria are prokaryotic cells that play an important role in human health and disease. They are part of human microbiota but can also cause certain diseases. Even though we can't see them with the naked eye, the biomass of the bacteria exceeds that of plants and animals combined. They can be found on our surface of our skin, inside our body, and also on every object around us. Compared to eukaryotic cells, the structure of bacteria is less complex. Bacteria's lack of nucleus and membrane-bound organelles such as mitochondria, endoplasmic reticulum, and also Golgi apparatus. There are two different types of bacteria, that is gram-positive bacteria and gram-negative bacteria. So most bacterial cells are protected by a cell wall composed of a unique molecule that is called peptidoglycan. Peptidoglycan is a complex monosaccharide linked by amino acids. We can differentiate whether the bacteria is gram-positive or gram-negative by gram staining. Gram-positive bacteria have a single thick layer of peptidoglycan above the plasma membrane, which allows them to retain the staining dye. This type of bacteria appears dark purple after the gram staining process, while the gram-negative bacteria have a thinner layer of peptidoglycan located in between the surface membrane and the plasma membrane of the bacteria, so they can't retain the color of the dye. This surface membrane blocks the antibiotic drugs, making infections difficult to treat. This type of bacteria appears red after gram staining process. You can try the gram staining virtual lab in the link provided in our description box. The cell wall of the bacteria also helps them to maintain their shapes. There are a few shapes of bacteria, such as cocci, bacilli, spirilla, and much more. Round-shaped bacteria are called cocci, rod-shaped bacteria are called bacilli, and the spiral-shaped bacteria are called spirilla. And sometimes bacteria can have multiple forms, and they are called pleomorphic. Bacteria can grow in different forms. They can grow in clusters, form chain, and also they can grow in pairs. When bacteria grow in clusters, they are described as the staph arrangement. For example, the Staphylococcus aureus is a bacteria that causes skin infections in humans. Next, when bacteria forms chain as they grow, they are described as the strap arrangement, as in Streptobacillus monoliformis and the Streptococcus pneumonia that cause pneumonia. Lastly, we have the diplo arrangement of bacteria, which happens when the bacteria grow in pairs. One of the examples of this bacteria is the Diplococcus bacteria.
Now, let's talk about bacterial metabolism. Metabolism refers to all biochemical reactions that occur in a cell or organism. Most bacteria are aerobic, and like animals, they also require a constant supply of oxygen to carry out cellular respiration. There are also bacteria called facultative anaerobes that can switch between an aerobic or anaerobic respiratory mode to generate energy, such as Salmonella and Staphylococcus sp. Some bacteria are obligate anaerobes and unable to grow in the presence of free oxygen. These organisms derive their complete energy from fermentation or anaerobic respiration. For example, actinomyces and Clostridium. There are also a few serious illnesses such as botulism, gas gangrene, and tetanus caused by anaerobic bacteria by infecting the oxygen-free region of human body, such as the small intestine or in deep punctured wounds. For autotrophic bacteria called photoautotrophs are photosynthetic. They use solar energy to reduce carbon dioxide to organic compounds. There are two types of photoautotrophic bacteria, which are those that perform anoxygenic photosynthesis and those that perform oxygenic photosynthesis. Their characteristics are shown in the table below. In anoxygenic photosynthesis, instead of water, they use hydrogen sulfide or thiosulfate as a reducing agent. They contain a photosynthetic pigment known as bacterial chlorophyll. Some of the examples are purple sulfur bacteria, purple non-sulfur bacteria, and green sulfur bacteria. For purple sulfur bacteria, they are found in hot springs and stagnant water, and they also thrive in anaerobic conditions. They utilize hydrogen sulfate or thiosulfate as a reducing agent. The main pigments are bacterial chlorophyll A and B, and they also uses a similar photosystem to PS2. For example, the bacteria Chromatia K, which gives the blood lake in Texas its blood-like color of water. For purple non-sulfur bacteria, unlike the purple sulfur bacteria just now, they uses hydrogen as their reducing agent. They are very important since they produce various beneficial substances such as polyphosphates and vitamins. For green sulfur bacteria, they can be found in the deep ocean in extremely low light condition. They contain bacterial chlorophyll C, D, E, along with bacterial chlorophyll A that can be found in the plasma membrane and chlorosomes. Their photosystem is also similar to PS1 in higher plants. In contrast, cyanobacteria or blue-green algae contain chlorophyll A and carry on oxygenic photosynthesis. They use water as an electron donor, and oxygen is produced at the end of the reaction. The reaction that occurs is as follows. Bacteria called chemoautotrophs carry out chemosynthesis, which utilizes chemical energy. They oxidize inorganic compounds such as hydrogen gas, hydrogen sulfide, and ammonia, which serves as a carbon source to obtain energy so that the reduction of carbon dioxide into organic compound can occur. For example, nitrogenous bacteria. They oxidize ammonia to nitrites, then nitrites to nitrates. This oxidation reaction leads to the release of energy. The converted nitrate is in turn utilized by the plants. Examples include Nitrosomonas and Nitrobacter. Next, let's move on to heterotrophic bacteria. Bacteria called chemoheterotrophs obtain carbon and energy in the form of organic nutrients from other living organisms, as they are not able to synthesize their own organic molecule. For example, parasitic bacteria that feeds on the tissues and fluids of their living host. Chemoheterotrophs bacteria called saprotrophs serves as decomposers that break down organic matter from the dead organism. Probably, no natural organic molecules exist that cannot be digested by at least one prokaryotic species, and this plays a critical role in recycling matter and making inorganic molecules available to the photosynthesizers. Saprotrophs feed by a process called absorptive nutrition, in which the nutritional substrate, for example dead organism or other non-living organic matter, is directly digested by a variety of enzymes secreted by saprotrophs. The metabolic abilities of chemoheterotrophic bacteria have long been exploited by humans. Bacteria are used commercially to produce chemicals such as ethyl alcohol and acetic acid. Even antibiotics are produced by some bacteria.
Biological interactions result when organisms in an ecosystem interact with each other. Despite their tiny size, prokaryotes can form close relationship with other organisms, which is called as symbiotic relationship. Symbiotic relationship can be divided into three types, which are mutualism, commensalism, and parasitism. When two species benefit from each other, the symbiosis is called mutualism. In human intestines, mutualistic bacteria will release vitamin K and B12, which can help produce blood components. In the stomachs of cows and goats, mutualistic bacteria will digest cellulose, enabling these animals to feed on grass. They also live in the root nodules of soybean, clover, and alfalfa plants, where they reduce atmospheric nitrogen to ammonia by a process called nitrogen fixation. Humans also have a mutualistic relationship with the bacterium Bacteroides micron, which lives in the intestinal tract. It will digest complex polysaccharide plant materials that human digestive enzymes cannot break down and convert them into monosaccharides that can be absorbed by human cells. In another type of symbiosis called commensalism is when one organism benefits while the other organism is unaffected. Commensal bacteria are found almost everywhere. They grow on animals and plants, exactly as they will grow on any other surface. For example, this occurs when the bacterium Staphylococcus epidermidis use the dead cells of the human skin as a nutrient. is a type of symbiotic relationship between organisms which the parasite will benefit from a long-term close association while the host is harmed. Parasitic bacteria cause disease and called as pathogens. The growth of microbes themselves does not cause disease, but they will release the pathological portion. When gram-negative bacteria are killed by an antibiotic, their outer plasma membrane releases a substance called lipopolysaccharide, which acts as a superintendent to overstimulate the immune response, which can cause a high fever and a severe drop in blood pressure, leading to shock and possibly death. The relationship between humans and many pathogenic prokaryotes can be characterized as parasitic. This is because the organism will invade the body and produce toxic substances that cause harm. Disease that cause from interactions between human and bacteria is tuberculosis caused by Mycobacterium tuberculosis, diphtheria caused by Corynebacterium diphtheriae, leprosy caused by Mycobacterium leprae, and pertussis caused by Bordetella pertussis. For example, when someone steps on a rusty nail, Clostridium tetani bacteria can be injected deep into damaged tissue and produce a toxin that will cause tetanus disease. The bacteria never leave the site of the wound, but the tetanus toxin produced does move throughout the body. This toxin prevents the relaxation of muscles and the body will contort because all the muscles have contracted and suffocation occurs. Some of the deadliest pathogens will form endospores when faced with unfavorable environmental conditions. In some bacteria, the rest of the cell deteriorates and the endospore is released. Endospores survive in the harshest of environments such as desert heat and dehydration, boiling temperatures, polar ice, and extreme ultraviolet radiation. Antibiotic is a type of antimicrobial substance active against bacteria and heavily prescribed to treat infection. Excessive and improper use of antibiotics will increase the bacterial resistance to antibiotics. Genes conferring resistance to antibiotics can be transferred between infectious bacteria by transformation, conjugation, or transduction. Antibacterial compounds can be divided into two classes: those that either inhibit protein biosynthesis or inhibit cell wall biosynthesis. Two types of antibiotics that inhibit bacterial protein biosynthesis by affecting bacterial ribosomes are erythromycin, that is used to treat pneumonia, skin disease, and sexually transmitted disease, tetracycline, that is used to treat pneumonia and other respiratory disease. Antibiotics that inhibit cell wall biosynthesis generally block the formation of peptidoglycan, which is a compound necessary to maintain the integrity of bacterial cell walls. Antibiotics that inhibit bacterial cell wall biosynthesis without harming the animal cells are penicillin, that is used to treat pneumonia, scarlet fever, and other respiratory disease, ampicillin, that is used to treat bladder infections, pneumonia, and gonorrhea, and lastly, fluoroquinolones such as ciprofloxacin, that is used to treat typhoid fever, infectious diarrhea, and urinary tract infections.
Cyanobacteria is a large diverse phylum in the kingdom of bacteria which consists of many species. What is special about the species is they are oxygenic phototrophs. Oxygenic means they are able to generate and release oxygen, meanwhile phototrophs refers to the ability of cyanobacteria to use light photons for nourishment. Putting it together, cyanobacteria use photosynthesis and generate oxygen as a byproduct. Cyanobacteria contain pigments and chlorophylls that are used for photosynthesis. Specifically, they have chlorophyll A and common pigment phycocyanin. Cyanobacteria also can have other pigments that mask the color of chlorophyll so that they can appear in red, yellow, brown or black color rather than only in blue-green color. Cyanobacteria also known as blue-green algae. But cyanobacteria are not algae, instead they are bacteria. This is because cyanobacteria don't have organelles like nucleus or chloroplast. This is contrast with algae, which is eukaryotic cells. This means cyanobacteria are prokaryotic cells. Cyanobacteria cell size 1 to 50 micrometer in width, and they have wide range of cell shapes and can live in single or in colonies. Some of them also are filamentous, forming long straight chains of cells. They have special cells mixed into the chain called heterosis. Heterosis captures nitrogen from atmosphere and transforms them into chemical forms to be used by cyanobacteria. This process is called fixing nitrogen.
Several species of cyanobacteria live symbiotically with other organisms, such as with fungi, ferns, and liverworts. In association with fungi, they form lesions that can grow on rocks, and lesions helps rock transforms into soil. Approximately 1.5 billion years ago, a cyanobacteria cell was engulfed by a primitive cell, resulting in a chloroplast, which is responsible for carrying all photosynthesis that can be found in plants and algae. The blooms of cyanobacteria is dangerous, as some of cyanobacteria produce toxic chemicals that can cause illness if being exposed to other organisms. Cyanobacteria are short-lived, so they will die in large groups. Floating mats of their dead bodies serve as food for oxygen-breathing decomposers who temporarily use up all available oxygen in water. When this happens, aquatic organisms like fish will die. Bacteria is one of the common genus in cyanobacteria. So since cyanobacteria doesn't have nucleus and chloroplast, their genetic materials mix in with the rest of the cell, and also the chlorophyll is located in their thylakoid.